So thank you. And I know I have a very long title and I'm standing between you and lunch, and the long title doesn't necessarily mean I'm going to run long. So there's my disclosure. I am a co-founder of Orlance. We'll talk a little bit about their technology. So this is our nucleic acid vaccine delivery platform that we use for delivering RNA and DNA vaccines. It's a gene gun that directly injects microparticles coated with either RNA or DNA into the epidermis. It results in very efficient delivery of the DNA or RNA. It's been actually tested in several human clinical trials, and because it efficiently directly delivers the nucleic acid directly into the cells, coupled with targeting the skin, which is a very immunocompetent organ, that it is much more efficient than other methods of delivering DNA or RNA where they primarily are delivered into the muscle. So as an example that we actually can see that in human trials, as low as 4 micrograms of gene gun delivered DNA will result in effective immune responses compared to 4 to 5 milligrams of DNA required, for example, by intramuscular injection with electroporation. The RNA that we use is actually a self-amplifying RNA vaccine, and that is very similar to mRNA, except that it encodes an additional gene that is a replicase that instructs that RNA to make additional copies of itself. And of course, when it makes additional copies, it makes more protein, and more protein results in a higher immune response. I'm going to mostly talk about actually the DNA delivery with the gene gun today. And so this is how this works, is that you can see that we actually deliver the microparticles directly into the epidermis. You can see nice gene expression in the epidermis, and this is what the particles look like in the epidermis after gene gun delivery. Once they go inside, you can see that the particles associated are directly transfect antigen-presenting cells, and then that actually launches our immune response. And so this is an example of a study that we did with a SARS-CoV-2 DNA and a rep RNA vaccines delivered by the gene gun in non-human primates. And you can see we administered different doses of updated SARS-CoV-2 vaccines. And what you can actually see here in 3 different groups, we have one group immunized with the gene gun with a self-amplifying RNA vaccine delivered, the gene gun delivery of the DNA, or we took that self-amplifying RNA and delivered it with a lipid nanoparticle intramuscular. And despite whatever route we use, you can see that we saw comparable antibody responses induced in all 3 of these different methods of immunization. And notably, they all induce comparable neutralizing antibody across different SARS-CoV-2 strains, including SARS-CoV-1 and a bat strain as well. So it also induced broadly neutralizing antibody. One of the things that we discovered about gene gun delivery is that when you immunize in the skin, it actually induces very potent mucosal immune responses. This is an example of a study we did in mice, which actually shows that when you compare gene gun to electroporation delivery of DNA, you get comparable gamma interferon T cell responses in the spleen. But when you look in the lung, you get much higher mucosal T cell responses with the gene gun delivery. We don't quite understand the full mechanisms of how that works, but we have found that when we look in draining lymph nodes, that we can find T cells have mucosal homing phenotype in them. In addition, if you actually pull out antigen-presenting cells from the gut or the lung mucosa, you can actually find APCs carrying skin-derived antigen located in there. So there may be multiple mechanisms playing a role for vaccinating skin inducing mucosal immune responses. And here's an example of that same study that we did with SARS-CoV-2 DNA vaccine and RNA vaccines in the non-human primates. You can see we get a really kind of reasonable T cell responses in the blood, but when you actually look in the lung mucosa, what you can actually see is the gene gun delivered RNA or gene gun delivered DNA induces really strong mucosal responses in the lung as compared to the lipid nanoparticle delivery of RNA, much higher mucosal responses. And so we began to think about whether we could leverage our nucleic acid vaccine technologies to develop a fungal vaccine, recognizing, and this was a surprise to me as a vaccinologist, that to date there's no such thing as a fungal vaccine, and we have vaccines for many other types of pathogens. And so what types of immune responses will this vaccine need to induce? Of course, we're all familiar with Th1 and Th17 responses are likely going to be very important for a vaccine. And this is what got me excited, because these are precisely the types of responses that both DNA and RNA vaccines are very good at inducing. Is there a role for antibody? Because DNA and RNA vaccines are very good at inducing antibody responses as well. The evidence suggests maybe not, because asymptomatic disease is generally associated with lower antibody, whereas severe disease is associated with higher antibody. And yet in mice, there is some evidence that suggests there may be a role for Th1-associated antibody responses. So there needs to be more research to determine if there's a role for antibody and protection. In my lab, we're very interested in the mucosal immunity because of the ability of our technology to induce really strong mucosal immune responses. And multiple studies that we've done with viral vaccines, we found that if you can induce tissue-resident T and B cell responses in the lung mucosa, that actually provides much better protection by blocking the pathogen at the initial site of entry. And so what we hypothesized is that we could use a gene gun delivered nucleic acid vaccine, either DNA or RNA, to induce protective mucosal and systemic immunity against valley fever. And so this is the first study that we did where we actually investigated DNA vaccines to start with, expressing immunogens that were known to provide some degree of protection when they were formulated as protein vaccines. We used the gene gun to deliver those into the skin. And what we did here is we actually co-delivered our vaccines with two important genetic adjuvants. And both of these genetic adjuvants we had tested with multiple viral vaccines before. And in addition, in human clinical trials, they've been shown to be safe and very effective. We used IL-12 because it's very effective at increasing gamma-interferon T cell responses. And we use a heat-label enterotoxin from E. coli because when you deliver that to the skin, it really augments the ability of DNA vaccines, our DNA vaccines, to induce mucosal immune responses. So here's our study design. I'm sorry that turned out blurry there, but we actually primed mice on day 0, and then boosted them on day 28. And then we shipped them down to Bridget Barker to challenge them on day 42 with 500 arthrochonidia in that. And so just to show you initially what the immunogenicity will look like is that the IL-12 and LT adjuvants were very effective in increasing the immunogenicity of our vaccines. What you can see here are the 3 different antigens that we tested. And for each of them, they induced decent responses, but we got a significant increase in gamma-interferon T cell responses when we delivered IL-12 alone or LT alone, but a synergistic effect when we combine them together. We looked at Th17 responses. We also see significant increases in both the Th17 responses, but those increases seem to be conferred primarily due to the LT adjuvant. Now, when we look at mucosal immunity, again, we're kind of seeing really very robust mucosal immune responses in these mice, and a similar pattern as we saw in the spleen where the IL-12 and LT are synergistic in inducing gamma-interferon T cell responses, and the LT adjuvant is really driving a robust Th17 response in the lung mucosa. We also saw very robust antibody responses. These are huge antibody titers, even without the adjuvant. When we actually co-deliver the adjuvant, and in particular the LT adjuvant, you see we get very high antibody responses against all 3 of these antigens. And so just to summarize what the immunogenicity looks like, with PMP1, that seemed to be one of our most robust immunogens. We saw very strong immunogenicity, both in gamma-interferon and Th17 responses, as well as antibody, both systemically and mucosally. We saw really robust antibody responses, especially for PMP1 and PRAAG2. And all 3 of these antigens really induced robust mucosal Th17 responses. So this is a challenge outcome, which was kind of interesting. Here, what we did is a DNA trivalent where we combined all 3 of these antigens, the PRAAG1, the PMP1, the ELI, together into a single immunization. And then we tested each of the antigens separately. We're comparing this to the TKO live attenuated vaccine and, of course, PBS naive controls. And what you can see is the trivalent DNA vaccine affords 100% protection from mortality and from morbidity. The PRAAG2 looks pretty good, almost on par with the trivalent vaccine. And we see modest protection from the other 2 vaccines that were actually on par or better than the live attenuated vaccine. When we dive a little bit deeper and look at fungal burden, what we can actually see is the trivalent vaccine actually is the only one that really significantly reduces the fungal burden in the lung and completely protects from a dissemination into the spleen or the brain. And just to remind you again what the immune responses look like, that PMP1 was the most immunogenic, yet it wasn't the most protective. And then all three of them were very effective in inducing mucosal Th17 responses, and it really was a trivalent vaccine that afforded the best protection. What was interesting is these two antigens also induces robust antibody. If you remember, the PRA-AG2 had pretty good protection, and so that began to make us think about, did antibody play a role in protection in this vaccine? And so we did another study where we investigated the trivalent vaccine. We primed and boosted the animals. And then one group of animals had a passive, we passively transferred the immune sera to naive mice. And the second set of mice had T cells, vaccinated mice got T cell depleted. And then we compare those to various controls. And this is what we saw. Here's the trivalent DNA vaccine control. Again, 100% protection. Here we're comparing it to the Delta CPS1 live attenuated vaccine, which performs pretty much similarly. But what's striking here is what we see is that when we do a passive transfer, we still see significant protection. Here we're looking at the trivalent DNA vaccine without the adjuvants, so we can see that the adjuvants were very important for the protective efficacy. We deplete the T cells, we still see some modest degree of protection, although we lose the majority of protection. And so together, this indicates that both T cells as well as antibody responses are likely playing a role in protection in this vaccine. And so just a brief summary here, the three adjuvant and DNA vaccines delivered by GeneGun induce robust antibody, mucosal, and systemic T cell responses. Combining the three immunogens afforded complete protection from disease and was comparable or superior to candidate live attenuated vaccines. And both antibody and T cell responses seem to contribute to protection. Previously, a role for antibody was not clear. So what's next for us? Now we want to look at whether we can incorporate additional immunogens targeting different stages of the fungal life cycle to further improve our DNA vaccine. But what parts of the cocci do we really need to induce immune responses against? And determining that can be a challenge. One way that we actually do immunogen finding studies is that you can actually produce proteins of different sort of antigens and then inject them into mice and challenge them and see whether or not they're protected. But that can take a long time just to produce those proteins. With DNA vaccines, it's really quickly to do this because you can bypass that step and actually just take the plasmids and directly inject them into mice. So in the next study here, what we did was we formed 24 DNA vaccines, each encoding a single novel antigen. Now, these antigens came from Bridget Barker, where she had identified them as candidate virulence factors in her studies. And so what we did was we formed 24 DNA vaccines, then we co-delivered 3 candidates just randomly into 8, divided them into 8 different groups of mice. And then we just challenged them feet up, feet down to see which of these groups afforded protection. And this is the result that you see. Now, I want to point your attention first to the controls. This VF1 here is our trivalent DNA vaccine. And what we did is, Bridget did, is use 1,000 CFU challenge, which is a very high dose. So we see the 100% protection isn't quite there, but it's still very highly effective. Here's the Delta CPS1. You can see that most of the protection is lost at this very high dose challenge, and there's a significantly better protection from the trivalent DNA vaccine when compared to the live attenuated vaccine. But what's really exciting is some of these candidate unknown or new antigens are actually affording a fairly significant degree of protection in several of the groups. You can see here 5 of the groups here had significant protection, and 3 of the groups we don't see any difference with the controls. And so then what's next? How do we actually identify potential candidate protective immunogens? So we first looked at protection, but we also applied concurrently additional criteria, and that is whether or not we see antigen expression and whether any of these antigens in these groups of 3 were immunogenic. And from those additional criteria, we can pull out 1 to 2 different additional antigens from each of these protective groups. And so the next thing that we're going to do is we're going to actually, it's ongoing right now where we've immunized mice with each individual antigen. We're going to see if each individual antigen alone can protect the mice. And then from those that we actually identify can afford protection, we can combine them with our lead trivalent DNA vaccine and see if we can improve protection further. And I just want to close my talk here to emphasize our earlier talk about using the pigtail macaque model to study coccidioides. Where we're going next is that eventually, once we have developed our optimized DNA vaccine, we're going to take that forward into a non-human primate challenge model. We are in the process right now of developing an experimental pigtail macaque model for coccidioides infection, so that it will be available to test not only our vaccine, but your vaccine and any of the therapeutics. We can also study these animals, which of course they're located right here in Mesa, Arizona, very exposed naturally exposed to valley fever. And we have a lot of macaques and historical specimens and repositories available if you're interested in studying immune responses or potential mechanisms of pathogenesis in this model. Those are available. You can go visit Megan Fredericks at Poster 18, where she can talk. She has a presentation about the status of our experimental challenge model. And that's it. Thank you.
Thank you. Because of time, I'm going to say if you have questions, come up during lunch.
Okay.
And
We're going to do it lunch.
Bridget Barker has some announcements.
So Janice reminded us about the pediatric meeting, room 161 for lunch. And there is a chance, so check the